Hi guys, this is Madhavji Padatil here for Sports to Science. Today we are going to be looking at lactate threshold and anaerobic capacity. So in today's session, we'll see how the physiology of lactate is, how is lactate produced, what are the type and how do we measure this lactate and what are the training implications of that lactate threshold. So lactate threshold, that is basically a point where the speed of lactate production increases than the speed of lactate clearance. So the body the amount of lactate produced is being higher than the amount of lactate that can be cleared. So here, the thing that we have to see is that VO2 max is not always the only way you can measure endurance. Lactate is also a very good way in which we can understand the endurance capacity. Here in elite athletes, they might reach lactate threshold at 85 to 95% of their VO2 max. Biochemical pathway. Now we are seeing how this lactate is produced. We know glycolysis, which is one of the very important way in which we produce energy. So glycolysis, we also know that it requires the least amount of oxygen to produce the most amount of ATP cells. Now during this glycolysis, the glucose is broken down into pyruvate at every cost at all in aerobic and anaerobic systems. But when it comes to anaerobic, where there is less oxygen available or limited amount of oxygen available, what happens is this pyruvate is converted into lactate using lactate dehydrogenase. So this lactate, or at that state it is called lactic acid, which is further broken down into lactate and hydrogen ions. So one important thing that we have to understand, this lactate is not uh, bad in any way. It is good. It is still energy producing cell. It can be very useful. So this lactate becomes a shuttle, helps to transport between cells, even goes to the liver where it undergoes Cori cycle, which basically breaks the lactate down into glycogen. This acid from the lactic acid, which the acid that is broken down, the H plus ion, that is a evil person here, which reduces the performance or increases the fatigue. Now defining LT1 and LT2. LT1 is basically lactate threshold 1 and LT2 is lactate threshold 2. So lactate threshold 1 is also known as aerobic threshold. So this LT1 and LT2 is kind of similar to where the body reaches VT1 and VT2. When it comes to when you're comparing it to work intensity or heart rate, it is mostly the same. So LT1 is aerobic threshold, where you see, as we told before, the body's ability to clear the lactate reduces and the lactate starts accumulating. So the first point where you start seeing the lactate increasing is known as LT1, where there is aerobic threshold. So after that point, what happens is the body is not completely going on aerobic mechanism. It is slightly also relying on anaerobic. That's why you start seeing the lactate being produced. After that, the LT2 is anaerobic threshold, from which point the body completely switches to anaerobic. There is no aerobic happening at all. So from there is a rapid increase in lactate in the blood. So 2 mmol is basically aerobic threshold, and that is 2 mmol. Just below the 2 mmol is the body's normal lactate levels. If it increases from there, you start saying, okay, there is anaerobic threshold. And this obla, onset of blood lactate accumulation, that point OBLA or obla point, is basically 4 mmol of lactate in the blood. That is a reference point that we see where the body will try to understand at which stage of the heart rate or which intensity of the body or intensity of their workload are they reaching their obla point, so that we can understand, and we'll try to manage the workouts according to those LT1 and LT2, so that we'll get a better results. Now, going to measuring methods. Basically, we can either do a lab test or a field test. In the lab test, it is basically an incremental exercise with lactate sampling, where in every stage you'll try to take the lactate to understand the amount of lactate, that is blood lactate. From there, we can understand it. Field test is basically concave test, so 30 minutes time trial. The math that it used is Dmax log or polynomial fits. So what happens is, from the fingertips or the earlobe, they tend to prick once so that one drop of blood comes. And most of you would have seen how glucose is tested. The same way, there is a strip which is connected to a device. If you drop that blood into that strip, you will basically get the amount of lactate in the device. So like that, you get the lactate at incremental exercise. If it's an incremental, it will be stage wise. Every stage, end of the every stage, you'll try to get that lactate to understand how the body is increasing that amount of lactate. Now, this anaerobic capacity and LT2. So this anaerobic capacity or VLA max, that basically is volume of lactate maximum. The maximum amount of lactate that can be produced in the body relates to glycolytic power, how much power the body is able to generate. So when you're thinking about that, this anaerobic capacity, the higher VLA max is, the more anaerobic power a person can generate. But one thing that we have to understand is here again, if the VLA max increases, the body reaches LT2 faster. It will reach. So that is something that we have to understand. Here, if glycolytic power is increasing, the endurance part will reduce slightly. So it is inversely correlated. And training adaptations. Okay, some important thing that we have to see is, now we talked about both the LT1 and LT2. So if a person now we know that, like, even if you're doing VO2 max test, or if we have the values of the ventilatory thresholds, here in lactate threshold we can basically set stages of workout using these lactate threshold values, so that a person can do workout in those stages, heart rate zones, so that they'll get their desired outcome. So for example, if a person has to improve their anaerobic capacity, it's better that they work on the final stage, or which is close to the lactate 2 LT2, lactate threshold 2 levels. So what are the major changes or adaptations that happen when you do at that stages? Endurance training enhances mitochondrial density. Obviously, the amount of mitochondria will increase. Then buffering capacity increases the body's ability to buffer the amount of hydrogen ions that are present. Because of this, lactate production also increases, and there will be increase in lactate clearance. The amount of lactate that is produced will be able to clear faster. So what happens is, you will gradually see a right side shift of the lactate curve. What does that mean? So the body reaches those LT1 and LT2 at a later stage of their workload or later stage of their heart rate, which basically says that they will be able to withstand the workload even more. They'll reach the fatigue even a bit more later. The time when the amount of hydrogen ions increase, or the amount where the lactate is in the blood and it's not being cleared, will move towards the right. You will be able to push a bit more. So here, this was a very brief idea of what lactate threshold is, how does it affect, and how in a very small way it will be the training will affect the lactate that is being produced. Thank you. I hope it will be helpful for you guys.